Hello, welcome back. Next up is our last panel, zooming on cancer.
Good afternoon. In the next panel, we will zoom in on cancer, a disease that still is a major cause of death, both globally and in our country. Recently, new therapeutic approaches have been improving the survival rates for this disease, and here we will take a look at some of these novel bioengineered therapies and diagnostic solutions. Our first speaker is a computer scientist and engineer with a 10-year experience in artificial intelligence. He founded Nilg AI, a consulting company in AI and data science whose work includes the use of deep learning to fight cervical cancer. Please welcome Dr. Kelwin Fernandes.
You can hear me well? Yeah. Okay. Okay, thank you. So, as you said, I will be talking about cervical cancer, fighting cervical cancer with artificial intelligence. This presentation was done with a major contribution from David Levitz from MobileODT, and we have been working on this together. So a little bit of background about how we came here. Well, MobileODT was founded in 2012 with the main goal of solving cervical cancer. So they built the first actual mobile and smart colposcope. Then in 2013, I started working with AI applications to cervical cancer as part of my PhD thesis. And in 2018, I founded Nilg AI, a consulting company in AI, and we joined forces with MobileODT in a collaboration to bring AI to cervical cancer and to finally solve this. And that is what brought us here to this talk. And just a small background about why cervical cancer. So the two main reasons to work on this area, the first one is that cervical cancer has a very high impact. Just to give you an idea of the impact of this disease, just during the time of this presentation, about 11 women will die from cervical cancer. That is kind of an entire football team, right? And which is more problematic is that most of these detections are between 35 and 44 years old. So we are talking about women in fertile period with small children, from small kids to teenagers, if we consider that the average age for first pregnancy is about 30 years old. So it's a major problem right now in the world. So the main reason to work on cervical cancer is because it has a very high impact. And second reason to work on this is because there is a lot of room for improvement. So what we can do can actually make impact in the life of 300,000 lives per year. So it's a major problem. So just to give you some ideas about this, so why I say there's a lot of room for improvement. Basically, from this 11-team football team of women, just one of them lives in high income countries. So 10 of them are living right now in low income countries. So there is something we can do there to take them to the level of these women that is living in this high income country. The second reason is that it's curable if we detect it early. So like above 90% of survival rates on 5 years if we detect cervical cancer early. So the main reason for these deaths is actually lack of screening. So if there is a lack of screening, and if we screen more and better, then we can solve cervical cancer. Cervical cancer is very special in the sense that it affects some endangered populations, so women that have been raped, assaulted, HIV+, refugees, sexual workers, prisoners. So there is a lot of very small and endangered communities that are seriously affected by this disease, and by properly tracing and taking care of these populations, we may have a major impact on their lives. So the two main reasons: there is a high impact in working in cervical cancer, and there is a lot of room for improvement. So we can see the progress of our work in a very short time lapse, because there is a lot to do at this point. Yeah? So if we detect cervical cancer in the first stages, we can almost for sure save those women. Why is still cervical cancer a problem? And the main reason is lack of ubiquitous screening programs. So most women aren't being screened right now, or aren't being properly screened and treated. And why is that? Well, the common guidelines for screening include HPV screening, which is HPV is the most common cause of cervical cancer, especially two types of HPV, but whatever. So the typical guideline is HPV screening plus cytology. Okay, and you can do this in two ways. You can either bring those screening camps to the women all over the world. You can imagine all this massive movement of doctors and clinical experts to all potential regions of the world to screen those women, or you can bring those women to the healthcare facilities. And as you may guess, if even in Portugal we don't have healthcare facilities everywhere, medical and family doctors for everybody, in these low and middle income countries, it's even worse. So most of these women are not being screened in their lifetime. So this is a major problem. After this HPV screening and cytology, or in some conditions at the same time or before, depending on the guidelines. Just a second. So let's say you may even deploy a cytology at a worldwide level. So the main problem with cytology, with this guideline, is that it takes, in countries such as the USA and UK and Belgium, so you can imagine like high-income countries with a lot of resources invested in healthcare, it takes like about an average of 6 days from the time you screen the patient to the time you get your outcome. So by that time, most of the women are already in their villages, and they don't come back to get the results or to get the treatment. So, and this is in USA, UK, Canada, and Belgium. So you can imagine in countries like Kenya, so they are already in their villages, they won't come back to the hospital because it's a major problem for them. So the second way of screening patients is with visual inspection with acetic acid, or colposcopy, which is basically visual observation of the patient. And in this case, we can observe what is going on with the patient, like in life, like in situ. Okay. The main problem with this is that we have these bulky devices. So you can imagine the size. There is a printer there for reference and a TV. So they are like huge. Okay. So you can do two things. You can bring women to these healthcare facilities that have a colposcope. Just to give you an idea, there are some countries that don't even have a colposcope in the entire country. Or you can take a very huge human to carry out these bulky devices all over the world to screen women in all these small villages and towns, and in very remote locations. So this is not possible, okay? And this is why most women aren't being screened right now. So how can we solve cervical cancer is the main challenge that brings us here. And our goal, Mobility and ourselves, is to solve one single need, which is screen and treat at the point of care, ensuring access to high quality care regardless of geography and other factors like cultural, age, whatever. What do we mean by screen and treat at the point of care? So when you look at when you get the chance to screen the patient, preferably where the patient is, not where the hospital is, where the patient is, you should be able to react. You shouldn't wait 6 days or even up to 3 weeks to react. You should be able to treat the patient, okay? And to do this, we need mobile devices, okay? We need mobile computing. We need to be able to diagnose or to screen the patient right at the point of care, not 1 week later or 1 month later. And the main issue to do this is that we need to scale up things, okay? And when we scale up things, when we try to ensure universal access to something, the tendency worldwide, and it happens with a lot of things like education, healthcare, or whatever, when you try to provide an universal service, by doing it in the same way that you're doing everything now, which is not changing the processes, what will happen is that you will start lowering down the quality bar, okay? And we need to make sure that we have universal screening, but at a very high quality care. And we cannot do this at this point because we cannot train humans at the same rate we can train machines, for instance. So the solution to have high quality care at a universal, at a ubiquitous scale, like everywhere, is by relying on artificial intelligence, okay? So we can ensure a minimum bar of what is the quality or the accuracy of the detection that you will obtain on those remote locations. So mobile devices, mobile ODT, I told you in 2012, started working on this issue by building the first mobile colposcope. And it's basically the size of a cell phone, a little bit larger because it has its handle, right? But basically, it's super mobile, so you can have it on your purse or whatever, you know. It's super mobile. It's functionally equivalent to a commercial colposcope, so to these very bulky and huge devices. It's basically comparable, it's equivalent in terms of its capabilities. And it's connected to the internet, which is a major advantage, and it's FDA cleared and it's being used right now at 42 countries. So it has a high acceptance. And the main advantage of mobile is that it blurs the geographic borders. Okay, it's not like you have this device here in this healthcare center and cannot be transported anywhere, so you can have it in your pocket. Okay, so the frontiers doesn't exist anymore. If it is connected to the internet, you may even have a doctor that is thousands of kilometers away from the patient and still be looking at the patient and giving his expertise to the case. So mobile mobile computing with connectivity is able to remove this constraint, and it allows teleconsultation. Mobile ODT also, besides the colposcope, they have a platform where doctors can submit the cases and get feedback from other experts in the area or have teleconsultation, whatever. So it's a very powerful device to make sure we can scale up these screening programs. And then the second thing we need, as I said, is artificial intelligence. Okay, because why? Because AI ensures a minimum level of performance, even if you don't have an expert at hand. So even if you have an untrained person examining the patient, you will make sure that the lowest performance you will get is the performance of the machine that is already validated. The second reason is that AI is reproducible. So every time you submit the same data from the same patient, you will get the same response. And being reproducible is very important in this area because you can audit the system. So you can know what happened on each stage and why this patient received this treatment or why the patient was asked to go to home or whatever. So reproducibility is not only important in research, it's important when we are building trustable guidelines for medical procedures. And AI can accumulate the expertise from all the patients all over the world. So it's not limited to the patients that you can see as an expert in your lifetime. So you can empower doctors with information they wouldn't get in any other way. So by traditional means, they will be, a Portuguese doctor will be able to get information from patients from China, from Africa, from wherever, because it's already all compressed into an AI model or into an AI framework. And AI can support experts in fatigue and burnout conditions because AI doesn't get tired, right? And this is super relevant when we are screening thousands of patients per day in these screening camps. The later the patient gets in the queue, the worse treatment or screening or the worse care she will receive because humans get tired. Okay, so small lesions can be missed, and with AI we can make sure that those lesions don't get missed just because the human is tired. So it's a pretty good way to suggesting the doctor, hey, you should be looking at this location right now and shouldn't miss this point. Okay. So what brings me here, I am not a medical doctor, I am a computer scientist, is the AI part. So how can we train AI models to detect cervical cancer? The first thing we need is data. And with mobility, we have a lot of data. I will risk to say this is the most comprehensive cervical cancer data set for ABE, because as you can see, this map is from all over the world. So we have data from all these points, biopsy validated data, 2,000 patients with biopsy outcomes, plus 100,000 patients without biopsy. So we have data from a lot of patients all over the world. And so this is not an academic data set. This is a real life data set. You have all the variability from untrained, trained physicians from all possible races you can imagine, skin colors, severity, whatever. So this is a very rich data set to train our models. And we are working on getting more biopsy validated data, which is the richest type of data, because it's the one with actual labels. And there are some open questions here that we should answer. The first one is, what data should we use? For instance, only relying on good quality images, or also including low quality images, because this is the real life setting that you will get in the field. How you should mix all this data from all these sources, ensuring that there is not bias on the data. How you can make sure the model is not relying on data from one place in the and not from the other. So how can you make sure all this data is treated by the model in a fair way? Is a very relevant question. And the second part is, what is the right ground truth for training the models? What is the label that we should use for each patient? We can either use biopsies, okay, or we can use human impressions. Each one of these has some pros and cons. For instance, with biopsy, we are considering data that pass the bias of the doctor for saying, hey, I think there is something suspicious here, so you should take a look and take a sample to this. So there is a bias there. But also with human impressions, we have a lot of noise from the humans, because it's very subjective. Okay, so there are pros and cons with this type of labels. The actual label we should rely on is if survival rate, but that is super hard to obtain. So this is an open question in cervical cancer. What is the right ground truth to train these models? And the second part to train this model is actually training the model. Okay, so you have the data now. What you should train your pipeline. We are relying on this pipeline. So the detection and cropping of the cervix, the data quality assurance, and the actual cervical cancer detection. We will talk about this a few minutes from now. So regarding the detection and cropping, we are using CNNs for bounding box estimation. Not much to say. Why? Because we want to remove all the external sources of noise, such as the speculum, the skin color. We don't want to rely on those regions, so we limit the field view of our later stage model to the region of the cervix. And then we have this quality assurance model that tries to give a threshold of whether or not the data is relevant for screening. So if we should rely on the outcome from this data, or if the data is so bad that we shouldn't rely on this. For this, we have a classifier that was trained with human annotations, so subjective quality annotations given by an expert, in this case by a committee of experts. But we want to go beyond this, because saying good quality versus bad quality is not very, it's not good in the sense of building better doctors, okay? It's just good in the sense of having high accuracy, but not on providing better quality access to healthcare. So we are trying to build models that actually guide the doctor on acquiring good quality data and giving feedback, such as there's a shadow here, so the model cannot observe this part of the cervix. You should try to adjust the position to remove the shadow, or there is bleeding, so you should clean out the blood to actually observe the entire cervix, or the camera is not very focused. So we want to train doctors and nurses to get better quality images, and this type of feedback is super relevant. So we are also working on building these models to provide user-oriented feedback. And if you want to know more about how to train a quality estimator, even if you don't have labels, I will encourage you to look at our at our blog. There you have the reference. Then the final step is actually detecting cervical cancer. And in general, when we talk about cervical cancer, we are not talking about actual cancer, we are talking about pre-cancer, okay? So we want to detect early stages, precancerous stages, but we will say cervical cancer, okay? So when you hear cervical cancer, we typically are talking about pre-cancer. And we have this scale, normal patients, CIN1, which is like a degree where the patient itself will probably get cured without doing anything. So typically these cases are, we will say, like, go home and come here 1 year from now, or whatever the guideline says. And the cases we actually care about is CIN2 plus. So yeah, so CIN2, 3, and cancers. And with these cases, we have a special treatment for them. And in this case, so we have a CNN, a convolutional neural network, or you have heard about it, and pre-trained on ImageNet. And then we train this CNN in a semi-supervised fashion. So we use the biopsy validated data in a supervised way to guide the model to detect CIN2 plus versus the rest. And we also use the unsupervised patients, which, as you saw, we have 100,000 of these, with small augmentations and ensuring the model gives similar predictions to the same image. Just for instance, we take the image, we rotate the image, and the prediction should be similar. Even if we don't know if the patient has cancer or not, we know the model should be consistent. So we combine supervised and unsupervised in this way. You have the reference there for the paper in case you want to know more about semi-supervised learning. Then we combine this with data augmentation at inference. So we take multiple images from the same patient and aggregate the prediction. So we reduce the variance of these models that were trained with relatively low number of images, 2,000 biopsies, 2,000 patients, not 1,000 biopsies, because we have multiple images from each patient. So aggregating the data from multiple data, so multiple images from the same patient, reduces the variance, increases the stability of the model. And also we have an ensemble of models. Just to give you an idea of the performance of these models, we are obtaining an AUC of 87%, which is quite high at this point, and considering all the variability that we have in the data. Then there is the next level feedback. So one is saying the patient has cancer. And this, for me, this is not enough. This is way better than what we have now. I mean, in most parts of the world, but we need more. And so we need to guide the doctor on where to take the biopsy, where is the lesion. So for now, we are doing like explainable AI. So we are trying to inspect the relevant regions to take the biopsy, identifying where are the lesions, etc. But what we want to do is build a lesion detection, okay, to guide the doctors on, hey, you should look at this region, not only saying cancer, non-cancer. So we don't want 5 years from now to only say cancer, non-cancer. We want to guide the doctor. So the ultimate goal in our case is also suggesting the right treatment. So not only saying cancer, not cancer, but what is the next step? So the patient should go home and come back in 6 months from now, or 1 year from now, or you should apply cryotherapy, or what type of treatment you should use with this patient. Yeah, so far, Mobility released this Kaggle competition together with Intel to detect the type of cervix, which is used in the decision of the type of treatment you should use. But in the future, we would like to have an end-to-end system that guides the doctor on where to take the biopsy, what type of treatment, what type of guideline you should follow, etc. So this is our ultimate goal. I told you this is not an academic project. This is a real-life project. So I wanted to share with you some results from our screening camps. Here you have some images from a screening camp in Nairobi, Kenya. And here's a fun fact about Afghanistan. Afghanistan, you don't have gynecologists, okay, only obstetricians. And there is, cervical cancer screening was non-existent there. They didn't have any access to screening on cervical cancer, and the EVA system, this mobile colposcope, was the first colposcope in Afghanistan. So this is the kind of impact I'm talking about. It's the impact from not having anything to having a mobile colposcope that can be everywhere in the world, especially in this case of Afghanistan. It's quite, it's a major thing. Another insight from a screening camp, this was in Inner Mongolia. We got Mobility got more than 3,000 patients. And this was the first use of AI in cervical cancer in low resource settings. So we are not working in silico in the lab. We're already there in the field, screening patients and validating the outcome of the algorithms. And so this is a major step. And these are the, just a second, I'm receiving a message. So this is the rock curve we got from the, getting just a message from the organization. I will just finish in 1 minute, one more slide, sorry. And I will just wrap up. So this is the curve we got, just to give you an idea. This is the cytology. So this is the level of cytology, of liquid bed cytology. As you can see, our curve is more or less in this region, a little bit above cytology. So this is, we are pretty sure our models are getting good performance on these patients. And as I said, liquid-based cytology gets like 1 week to get results. This is instant access, okay? Yes. So current guidelines put colposcopy in the end. First, we have HPV and cytology. As I told you, this incurs in major delays for screen and treat. What we want is to have AV on top of this. So as the first step, so you can have screen and treat at the point of care, which is so we don't want to miss patients and let them be at home while we actually can diagnose them at the right minute, at the right instant, they pass the door. Okay, so just a final outcome we got, and this was very interesting. I observed this last year when I went to Kenya to a few hospitals where they are using a mobile ODT device. Women which are shown images of their cervix are kind of more responsible. They feel more responsible for the outcome, so they are more likely to adhere to treatment. And you can feel this when they see the images from their cervix. They kind of feel responsible for their health, so they are more likely to adhere. So forget about the AI, forget about anything. Just looking at the picture makes them be responsible of their future and to adhere more to the treatment. So just a key challenges to address from healthcare. We have all the next steps in policy making and building the right ecosystem for universal screening. From an AI point of view, we need to make sure we have the right data and the right labels. We are not sure what are the right labels at this point. We are using biopsy, which is a pretty good source of expertise, but the right label should be saving people. And we are not sure what is the right label that makes machine learning save people. We are working on that. A final slide with some acknowledgements for the team working on this. Thank you all. Thank you.
Thank you, Dr. Kelvin. Our next speaker, Flávia Sousa, is a research associate in the Department of Materials of Imperial College London and holds a PhD in Biomedical Sciences from Universidade do Porto, having received several awards for her work in nanocarriers for cancer treatment. Please welcome Dr. Flávia Sousa.
Flavia, can you please turn on your microphone and your camera? Flavia, can you please turn on your microphone and camera? Okay.
Yes, it's working, right? Okay. So before I start, I'd like to thank the opportunity to share my work here with you and thank for the invitation. And I'd like to present myself. So my name is Flávia Sousa. I'm a research associate in the Department of Materials at Imperial College London. And today I will talk about the work that I did during my PhD in Porto at I3S, which is about the PLG nanoparticles for the delivery of antibodies in the treatment of a glioblastoma, which is a brain cancer. So it's quite far away from the previous speaker, but it's fine. So I will start to give a brief introduction about this nanosystem, because my expertise is more or less about drug delivery using nanoparticles. And why I'm so interested in nanoparticles, because we can achieve, the nanoparticles can give us some advantage, such as like the control release of the drug, because some drugs have short half-life, so we need to improve that half-life, and we can achieve with nanoparticles. Otherwise, we can also target a drug to a specific organ with the called active targeting. So with this case, I will turn this here. So you can put in the surface of your nanoparticles some antibody proteins, or also some cell-penetrating peptide, and with that way you will direct the nanoparticle for your specific target, your organ that you want. On the other way, these nanoparticles can be made by organic or also inorganic polymers. So, and one other advantage that we can get with nanoparticles is the intracellular delivery of these nanoparticles. Basically, you have your drug inside the nanoparticles, and the nanoparticle will be internalized in the cell, and the drug will be supposed release in the cytoplasm. One of these right now a hot topic is for the coronavirus disease. Actually, you can deliver the mRNA inside the cell, which is actually it's a hot topic right now, right? And at Imperial College, there we are working on that as well, either for the treatment with mRNA and also for the diagnosis. So this is just one thing that you can get with this nanosystem. And also one thing that you can have is the permeability of these nanoparticles through the epithelium, and as it is dependent of the particle size of these particles. So you can see here, if you achieve nanoparticles between 200 and 500 nanometers, you can have high permeability of these nanoparticles, depending, of course, on the source of the nanoparticles. So this was just a brief introduction, and right now I will talk about the cancer. And during my PhD, I was focused on the glioblastoma, which is a brain cancer. So this glioblastoma is a very aggressive cancer, which has a lifespan of 50 months after the disease diagnosis, and it is the most common and fatal form of a primary brain tumor. On the other hand, the glioblastoma is among the most vascular cancers due to the high rate of the blood vessels in the brain. So one of the treatment right now for this glioblastoma is the use of the bevacizumab. Bevacizumab is a humanized anti-VEGF monoclonal antibody, and it is responsible to bind to the VEGF protein, avoiding the angiogenesis and also, of course, the tumor urogenesis. And currently, bevacizumab is used as a single therapeutic agent to treat the glioblastoma with progressive disease as well. So, however, there are some studies really about the limitation of the glioblastoma treatment with the bevacizumab, because the bevacizumab itself presents limited BBB crossing and also has some off-target toxicity. On the other hand, there are described some studies which reports the cancer resistance of this bevacizumab, because the own cancer start producing some extracellular VEGF, so it's a cycle. To overcome all these limitations about the bevacizumab treatment, what we thought was to use the drug delivery and nanosystems as nanoparticles. Why? Because we want a controlled release. We want also to overcome the DDB, since it is a brain tumor and we need that. And we also want to achieve an intracellular monoclonal antibody delivery. Why? Because the own cancer cells start producing VEGF, so the own cells has intracellular VEGF, and also we want, of course, to reduce the off-target toxicity that is presented. So what we thought was to encapsulate this bevacizumab into inside the PLGA nanoparticles. And why PLGA? So PLGA is a copolymer, and it is biodegradable, biocompatible, and it presents low toxicity. But the great advantage is there is FDA approved. So currently there is some medicines already approved by the FDA, which are composed for this with PLGA. So that's why we choose this polymer. So I will show right now some of the results that I obtained. So this is the characterization of the nanoparticles. As you can see here, the nanoparticles present a particle size at around 200 nanometers. They are quite homogeneous with a low PDI. They are also negatively charged. And here you can see the spherical shape of these nanoparticles, and on the right side you can see the bevacizumab release from these PLGA nanoparticles. And as I said previously, for instance, after 1 week, the bevacizumab was released more or less at around 14% from these PLGA nanoparticles, so which is quite good, and it's what we wanted at that time. Then we move forward, because bevacizumab is a monoclonal antibody, so we need to guarantee that its structure doesn't change over time. So what we did was some biophysical studies regarding the bevacizumab structure. Probably all of you know. Basically, we used FTR, CD, fluorescence spectroscopy, and we didn't find any changes in the bevacizumab structure before or after the encapsulation. We did also a 6-month long-term stability of these nanoparticles, because we aimed to deliver the nanoparticles in vivo models. So we did this study, and basically we found a formulation that was stable over 6 months for either the bevacizumab structure and also for the nanoparticles. So we move forward with this formulation, and we study the bevacizumab bioactivity after the encapsulation. So as I previously said, the bevacizumab binds to the VEGF, and the angiogenesis will decrease. So here it is MTT reduction assay, so we are looking for the viability, and as you can see, after the bevacizumab here, the viability of the cells decrease, and also the loaded PLGA nanoparticles, so which is good, and we could say that the biological activity of the bevacizumab was maintained after the encapsulation of bevacizumab. Then we move for the in vivo efficacy studies, because as I said, we want to deliver these nanosystems in a, I don't know, clinical trials, phase 1 or something, and we need to do this in vivo study. And for this, what we observed was the tumor growth and also the angiogenic profile, because as I said, bevacizumab is an anti-VEGF, a monoclonal antibody. So the first study that we did was the in vivo efficacy in a zebrafish model. And just for basically we inject the U87 cancer cells, which are derived from the brain, and we inject these cells in the transgenic zebrafish, expressing GFP in the endothelial cells. Why GFP in the endothelial cells? Because we wanted to observe the angiogenic profile and also the tumor growth. So the tumor cells were marked here as a pink color and the endothelial cells by green. So basically after 48 hours post injection, we could see the tumor inside the zebrafish, which was good. And then we move for our treatment control. So here you have the free bevacizumab, loaded pills and nanoparticles, and also the empty, just the control, of course. So we evaluate the tumor area after the 48 hours post-injection, and impressively we saw just a decrease in the tumor area for the group of bevacizumab loaded pills and nanoparticles, which was quite good. And then we observed as well the vascularization index, as I said previously, and we observed a decrease in this vascularization index either for the free bevacizumab and also for the loaded pills and nanoparticles, but the decrease was higher for the group that for the group of the loaded pills and nanoparticles. So our hypothesis was maybe the pills and nanoparticles are internalized by the cancer cells and in some way can be interact with the intracellular VEGF. So our next experiment was basically the same as previous, but here we use the same control groups, the same cells, but in vitro, and we quantify the number of bevacizumab positive cells and also the number of VEGF producing cells. And what you can see here is with the loaded pills and nanoparticles allowed a higher internalization of the bevacizumab, and also a decrease in the number of VEGF producing cells, which means that this kind of nanoparticles might cause a disorder in the VEGF signaling pathway. So which was what we want, of course. And then we move for the in vivo in mice model. Okay, so basically the procedure was the same. We inject the same cancer cells in nude mice, but in this case we use luciferase cancer cells just to observe in the IVs. And after 10 days we are able to see the tumor, as you can see here, in the mice. So our therapeutic plan was we inject, so at day 0, these cancer cells. At day 10, the tumor model was complete, so we were able to administer the free bevacizumab and also the nanoparticles, and we did this by the intranasal route. Why the intranasal route? Because we can overcome the BBB, because there doesn't have. So, and we did also one more administration 1 week after, and after 2 weeks we observed the tumor growth and also the angiogenic profile. So here what you can see is after 2 weeks of treatment we observed a decrease in the tumor growth, either for the group of free bevacizumab and loaded pills and nanoparticles, but we didn't find any difference between these two groups. But we observed as well the angiogenic profile, which was what we wanted, and we observed a decrease in the VEGF, in amount of VEGF in the brain, regarding the bevacizumab loaded pills and nanoparticle, which was what we wanted at that time. And I also want to say one more thing, which is okay, this I don't remember. Okay, let's move it because I forgot. Okay, as a conclusion, we were able to encapsulate the monoclonal antibody into polymeric nanoparticles, PLGA specifically. These kind of nanoparticles were not functionalized with either a protein or antibody or whatever, because we use the intranasal route. Okay, so we basically overcome the BBB. And we found a stable nanosystem over 6 months that allowed the slow release of the bevacizumab. We also achieved intracellular delivery of this monoclonal antibody, a decrease in the tumor growth in vivo, and also a higher anti-angiogenic effect of in the glioblastoma. So I'd like to acknowledge not my current work, because this work was done in my previous during my PhD. So I'd like to thank Bruno Sarmento for the opportunity to do this job at I3S in Porto, and also some of my collaboration at INL, CESPO, Aalborg University in Denmark, and Leslie in Boston, so at Northeastern University with Dr. Masarami G, because the in vivo work was done there. So that's all. Thank you so much.
Thank you, Flavia, for your amazing presentation. Thank you, Flavia. Now I will ask Kelvin to please turn on his microphone and camera, and also you, Flavia, so that we can have a little debate and answer some